Welcome to our presentation of the CAMAG HPTLC PRO, the first fully automated system for high-performance thin-layer chromatography. My name is Melanie Brossard and I am the Scientific Business Development Manager at CAMAG since 2014. First, a few words about our company. CAMAG was founded in 1958 in Switzerland, and the first product was aluminum oxide for preparative chromatography. This product is still sold. Since 1961, CAMAG is dedicated to thin-layer chromatography, TLC, and from 1975 on, CAMAG concentrated its activities on the development of instruments and automated solutions for TLC. Since the mid-80s, CAMAG provides software, so our current software is called VisionCats, and the software allows to control the automation of TLC steps and to evaluate the results. Last year, in 2019, CAMAG has launched the first two modules of the fully automated HPTLC PRO system, and the remaining modules will be launched sequentially over the next 2 years. In addition, CAMAG provides an automated high-throughput Elution instrument, the DBS MS500, to the clinical diagnostics industry. The DBS MS500 allows to automatically elute spots from tried matrix spots filter cards. CAMAG is present worldwide. The headquarters are located in Switzerland. Then CAMAG has two subsidiaries. One subsidiary is located in Germany and the other one in the US. And CAMAG has more than 60 distributors serving more than 90 countries worldwide. Before I will present the HPTLC PRO, I would first like to give you to explain the difference between TLC and HPTLC and introduce you to the concept of standardization. Every chemist knows the manual TLC, which can look like these. Not everyone is familiar to high-performance thin-layer chromatography, short HPTLC. So with a manual TLC, you can get fast answers about is there a zone present or not. It gives an idea about a sample composition, but the results are only comparable on the respective plate. Each plate will look a bit different from the RF values and also from the colored zones after derivatization. HPTLC, on the other hand, gives valid analytical results. It's reproducible, and therefore the results are comparable even from different plates. To get reproducible results, several requirements have to be fulfilled. For HPTLC, suitable instruments, a scientific basis, a standardized methodology, validated methods, and a system suitability test to qualify the data are required. CAMAG offers a software that supports a standardized methodology according to regulations, and also it allows you to be in full compliance with 21 CFR Part 11 requirements. With a standardized methodology, results are comparable from different plates, and with the software we can create a, let's say, virtual plate that is generated with a comparison viewer. Here you can see 4 different plates, and with the comparison viewer you can select tracks from the different plates and put them side by side on a virtual plate. Then they can be compared to similarities and differences. So with HPTLC you can cluster sample tracks if they look similar or different and see which spaces, which plant species was used or which plant species is present. HPTLC is the visual chromatography that also allows you to easily detect adulteration of plant materials with other plant species. CAMAG has 3 segments for TLC and HPTLC. So for TLC we have devices that are used in R&D, QC and mainly for qualitative tests, also for education purposes. Then we have the HPTLC segment with instruments for qualitative and quantitative analysis. Can be used in R&D, QC, education and small-sized companies. And now we have the HPTLC PRO, the first fully automated sample analysis and evaluation system for routine quality control. This is addressed for routine labs that have a high sample throughput. All the 3 segments are supported by the VisionCAD software. So the software also allows you to document manual steps, to have no gaps in your method. And when you create a method, you just click on the icons in the sequence that is required for your method according to the SOP. Therefore, it's also possible to have a mix of existing HPTLC instruments with the new HPTLC PRO modules. The VisionCAD software is easy to handle and it will guide you through the analysis. And at the end, it allows you to evaluate your data, to do a reliable quantification, to compare profiles, and also to compare spectra. The data can be acquired by image documentation or by scanning densitometry, and soon for the HPTLC PRO by hyperspectral imaging. Hyperspectral imaging means that you get in each position of the blade the entire spectrum from 200 to 1000 nanometer. The VisionCAD software also contains a method library, and each entry in the method library consists of an instrument method that is ready to use, a describing method document, and a comparison viewer file with sample image tracks that our lab has generated. With this comparison viewer file, you can import your own data and compare them to the results that we have obtained. So what are the benefits of HPTLC? First of all, HPTLC is the visual chromatography and very flexible in use. Then HPTLC is a reproducible analytical technique and gives you reliable results. You can reliably quantify. Then, what is really a big strength of HPTLC is that once analytes have been separated, you can then detect them not only once, you can detect them multiple times. In this example, you can now see samples originating from different blades and then detected at different stages. So prior to derivatization under UV254 and the native fluorescence under UV366, then after derivatization under white light and under UV366. One separation for each sample, but multiple times detected. And multiple samples can be analyzed in parallel without cross-contamination. Each sample gets a new position on a plate, and there cannot be any cross-contamination from a previous sample. a substance confirmation is required, then HPTLC can easily be coupled to mass spectrometry by the use of the TLC-MS interface or the module MS interface for the HPTLC Pro. You just elute your zone of interest and immediately get your mass spectrum. The HPTLC process consists of several steps. The major steps are application, development, derivatization and detection, and if required, also coupling to mass spectrometry. The coupling is rapidly done, so you can connect the MS interface with your LC pump and your mass spectrometer that you use for LC-MS. It's like an add-on, and then you can select the zone of interest, localize the elution head above this selected zone. The LC pump will dissolve the sample zone, and then the filtered zone will be transferred to the mass spectrometer and you get your result. So the future of HPTLC has begun. Now we have the CAMAG HPTLC Pro, and also this HPTLC Pro system consists of individual steps. It's a modular system. We have a module for application, development, derivatization, detection, and for the hyphenation, the MS interface. CAMAG HPTLC Pro stands for full automation, leads to the highest reproducibility possible for HPTLC, and supports high sample throughput. Within an analysis, there is no manual intervention required because the plate is transported from one module to the next one via the conveyor. The plate is held by a carrier, and this carrier is recognized by the system, so at any time point during the entire process the location of the plate is known. This is for full traceability of cGMP. Here a short teaser video. So the plate is loaded, then it's applied with a laser controlled distance, gets into the developing chamber. If needed, it is derivatized. Then the plate is detected by hyperspectral imaging. And if needed, a substance confirmation by HPTLC-MS is performed. So the first two modules launched were application and development last year. And what is new for application, for the module application, is we have the laser controlled needle distance. So always the distance between the blade surface and the needle is kept constant. We have a precise application of needed volumes. For application, only the required volume is filled into the syringe, and it's always quantitative. There are two rinsing solutions to have a proper cleaning of the syringe. And in the syringe is a separation bubble to prevent cross-contamination. Up to 75 samples can be applied on 5 HPTLC glass plates in the format 20x10 without any manual intervention. The module comes with a 10 microliter syringe that has a serial number, and from outside all modules look the same. They have the same design, and on top of the application module you have the 3 bottles. One is for the waste, and then you have 2 for the rinsing solutions. Each module comes with 2 carriers to have the blade loaded. And for the module application, it is also delivered with a rack. Then what is new about the development? So for the module development, we have a low chamber volume to better control the gas phase. The distance between the layer of the stationary phase and the chamber wall is very small, which speeds up the conditioning processes. Therefore, we have a faster activation, and also the preconditioning of the stationary phase is speeded up. This is also supported by a circulating gas phase. The gas phase is controlled during development, and also we have a control of the volume of the developing solvent during chromatography. The solvent level inside is kept constant during the development process. We have 3 solvent bottles that allows you to have an automatic change between developments of the developing solvents. So the major advantage here is that we keep the gas phase under control, and that we have now a gas phase, a circulating gas phase, that can be used to further optimize the separation. On top of the development module are 5 bottles. So we have the waste bottle, the bottle for the activation, the humidity bottle, and 3 solvent bottles for up to 3 different developing solvents. So the gas phase can be actively circulated prior and during chromatography, and this is one of the real innovations for HPTLC Pro. This was not possible before. The gas phase is generated in the conditioning tube, and this can be generated from the same or from a different developing solvent, allowing to also change the selectivity during chromatography. So even during the development, the gas phase can be circulated. For those who have methods already developed with the twin draft chamber or the ADC2, the methods can be transferred from those chambers to the HPTLC Pro Module Development. On the left hand is an example on normal phase, and on the right side an example on reversed phase. So in the ADC2, the standardized methods require an activation at 33% relative humidity and then development in a saturated chamber with filter paper. The activation is still needed for the HPTLC Pro, but then we adjust the chromatography either by preconditioning or conditioning during chromatography, or by a combination of both. So on normal phase, when we use the same solvent for preconditioning and conditioning, then you can adjust the position of the RF values in the chromatogram. The separation will not change, but the elution strength, that you have either higher or smaller RF values. Preconditioning will usually decrease all RF values, and conditioning will increase the RF values. In the instruction manual, there is a scheme how you can proceed to transfer existing methods. But if we use for preconditioning or conditioning different solvents, then we can also change the selectivity and then further optimize the separation. End of this year, 2020, we will launch the module blade storage, and this is needed to automatically feed the system. So with the HPTLC Pro Module blade storage, 5 clean plates can be loaded in one stacker, and then up to 5 analysis files can be submitted and are then automatically processed. The processed plates are then stored in a separated second stacker in the plate storage and can then be further processed manually. As mentioned, the additional modules, the derivatization, MS interface, and detection will be launched during the next 2 years. So for HPTLC Pro, we have several target groups, but in general, HPTLC Pro is addressed to routine QC laboratories with a large number of samples per day. Can be limit test in pharma, can be for food testing labs, contract laboratories providing analytical services, and for all companies that need a fast screening method to analyze multiple samples, and especially those that have a diverse, complex matrix. One example that is also of interest is the limit test for the hemp industry. HPTLC allows you to screen multiple samples in parallel, and then with this efficient screening you can say is the total THC content above or below the set limit. So now I would like to introduce you to the concept of comprehensive fingerprinting. Up to now, HPTLC is mainly used for identity testing in the botanical industry, but the fingerprint also harbors information on the purity and the content. How do we generate data in HPTLC? First, we extract the sample, we apply, we do the chromatography, and then we generate the data. And data can be generated multiple times prior to derivatization, after first derivatization, and in some cases we can also do a second derivatization on the same plate. And then we use this set of data and do the evaluation. So the HPTLC fingerprint contains information beyond identity. We get from one sample the images, the peak profiles from images, the peak profiles from scanning densitometry, and soon also the profiles from hyperspectral imaging. Hyperspectral imaging will be somehow the combination of images plus profiles with the spectral resolution. And with these acquired data, you get a lot of information about your sample. In this animation you see one separated sample and detected at different stages, prior and after derivatization with different detection modes. So this is a very strong tool for clear identification, and by the intensity and position of the zones you can also get the information about the content. The multidimensional data analysis allows you to set really unique acceptance criteria for each sample. And this is our vision, that the software will do the pass or fail test without requiring the human interpretation of the data. Not completely, but that first the software will tell you the sample is okay or not. And for those that are not okay, you can further treat the data and see what is the issue with the sample. So we have the multidimensional data of one sample acquired, and then we can define our acceptance criteria that are like a template. And our sample has to match in this template with the number of zones, the intensity as well. If there are additional zones, then it will not pass. Here is an example of comprehensive HPTLC fingerprint from Deborah Frommenwieler, who is working in our lab, and she did it together with other partners. This is the case study of Angelica gigas. So with the ID method, she is able to distinguish 28 Angelica and related species. And the same method can be used to test the purity. Is there adulteration with a related species? And the same method can be used to set quality criteria, a reference standard that is used to check for the minimum content that the product has to have. Another example for comprehensive fingerprinting is here this Ginkgo study. So we have one method that tells everything. On the left side you can see here in the fingerprint of the reference substances, the system suitability test, then a Ginkgo extract reference material, and several Ginkgo products. The first two products, they are good, and all the others are not. Either the fingerprints are weak, or they have additional zones of added flavonoids, or additional zones of added other plant material. So for example, the products 5, 6, 7 and 8 have additional greenish zones that detect the presence of Sophora fruit. And for track 9 and 10, there can be seen a very weak fingerprint or even no fingerprint, and two strong zones originating from rutin. In those products, just the rutin was added, and the total flavonoid content test will not show this. It will pass in the cuvette test, but it will fail here in the fingerprint because the fingerprint shows there is only intent zones of rutin and not the presence of the Ginkgo, of other Ginkgo components. The whole study was published in Journal of Ethnopharmacology and also printed in our CBS customer journal. The method allows to do the identification, check the purity, is there the presence of other plant materials, and it allows you to perform a limit test to detect the addition of cheap flavonoids because of economically driven reasons. And another example of efficient HPTLC testing or efficient screening is cannabis testing to check the total THC content. For the content and for the limit test, it's just needed to have application development and detection. So for the limit test, then also you can do a precise quantification by multi-level calibration, and you can analyze your target or confirm the presence of your target analytes by UV spectra. If you add two additional steps for derivatization, the derivatizer and the documentation step, then you also get the fingerprint, the visual fingerprint, that you can use to check the identity. Is it medical hemp or is it industrial hemp, and which major cannabinoids are present. So now just a few words about other options that you have with HPTLC. Besides the classical HPTLC, it's also possible to couple HPTLC with effect-directed analysis. After separation, the solvent is evaporated from the plate, and what is left is the plate and the separated substances. That allows you to apply enzymatic solutions or bacterial cultures or yeast cultures, do the incubation, and then the readout. Then you will not get just the information about the physicochemical properties of your sample, you will also get the information about which substances have an effect. Common assays are listed here. So of interest at the moment is, for example, the Planar Yes estrogen screening to screen for estrogen active compounds on non-target screening, and the Salmonella typhimurium for the UMU test to screen for genotoxic compounds in, for example, food packaging materials. A possible setup for effect-directed analysis can look like this. This was a setup to look for bioactive products for the pharmaceutical industry in marine sponges. So the beginning is the same. We have the application, development and detection, and then we apply our bioassay to the incubation and then the readout, and active zones, they are eluted and further analyzed by high resolution MS or NMR or infrared. CAMAG has a lot of information on the website. So we have our customer magazine CBS that is released twice a year, and in there are application examples from customers in our laboratory. Then we have the literature database CCBS that has more than 11,000 abstracts, and you can search within free of charge. Each abstract gives you the information about the chromatography or about the HPTLC methodology, how was applied, which plate was used, which mobile phase was used, was there derivatization needed, and how was the plate analyzed. Then we have videos on YouTube and LabTube, so our channel is called CAMAG.com. Please have a look at our tutorial videos and on our product videos. And if you have questions, then please write an email either to myself or to sales@camag.com. Thank you for your attention.